Okay, so welcome everybody to a new round of Global IMMUNOTALKs. If this is the first time you're joining us, just to very briefly remind you that the goal of this series is to bring immunologists across the world to really top seminars in an egalitarian manner, and to do this without the need of traveling. Today is a special day because we are starting our second series of talks. The talk is going to be given by Carola Vinuesa. Carola lives in Australia, and so these global initiatives sometimes requires that people get together from many different places across the world. So for this talk, we connected with Carola back on Monday. It was actually Tuesday in Australia. It was 10 a.m. In Australia on Tuesday, but it was still Monday in the US. It was 8 p.m. in New Haven, and I think 5 p.m. In San Diego, and the three of us connected, and we have a pre-recorded talk from Carola, who of course cannot join us today live, as hopefully she's sleeping right now. But with this, I will then ask my friend Elena to start broadcasting Carola's fantastic talk. I'm sure you're up for a treat.
Perfect, Carly, thank you so much. So I will share my screen now, and I will, without further ado, I will start then the pre-recorded session. So you should see now black, and I will start it now. So it's recording. So hello everyone. As you know, this is the pre-recorded session for the Global IMMUNOTALK that is scheduled for Wednesday, August 12th by Dr. Carola Vinuesa. And it is my real pleasure to introduce to you today Dr. Carola Vinuesa, who is full professor of immunology at the John Curtin School of Medical Research at the Australian National University in Canberra. So Carola is originally from Madrid, Spain, where she received her Bachelor of Medicine and Surgery from the Autónoma University of Madrid in 1993. She later moved to the UK, where she received her master and PhD degrees in immunology at the University of Birmingham, where she studied the biology of B-cell germinal centers with Dr. Ian McLennan. In 2001, Carola received the Wellcome Trust Visiting Research Fellowship to perform her postdoctoral training at the John Curtin School of Medical Research at the Australian National University with Dr. Christopher Goodnow. And then she became a research fellow and the head of the Department of Pathogens and Immunity, and later on the head of the Department of Immunology and Infectious Diseases at the same institution. So Carola has a long list of very prestigious awards and honors. Of course, it would be impossible to name all of them in a short period of time, but I would like to mention that in 2008, she was named the Australian Life Scientist of the Year via the Science Minister's Prize. And in 2009, she was named the Gottschalk Medal by the Australian Academy of Sciences. And in 2015, she was elected Fellow of the Australian Academy of Sciences. She has a large number of publications, over 110 publications in top-tier journals, and she has been ranked by the Web Sciences as the 2019 highly cited research. As you know, Carola has tremendously contributed to advancing our understanding of how our immune system produces high-quality, long-lasting antibodies in both the context of infectious and autoimmune diseases. Her team was one of the groups that identified the follicular helper cells as a distinct subset, and also identify key regulatory molecules that modulate the follicular helper cells, and show that these defects in TFH regulation can result in autoreactive B cells and autoimmune diseases such as lupus or type 1 diabetes. More recently, Carola has identified gene variants that associate with autoimmune diseases in humans, and I'm sure we will learn more about her work today. Last, I would like to mention a little bit of a personal note and share with you that is very, very special to host Carola as a global immune speaker today. I met her when I was an internship in the School of Medicine in Birmingham. I was in the middle of my PhD training in Argentina, and I had an internship in UK. And Carola was at that time at the end of her PhD. She was preparing to move to Australia for her postdoctoral training. And she was extremely welcoming with me and my family during that time. And I will always be deeply grateful for her tremendous generosity, not only to share her brilliant thoughts to advance my scientific project, but also offering her advice on how to move forward to the next step of my career. And so this very early advice was of great value and impact for me. And so hosting Carola today is not only a great honor, but also is a personal delight to have you with us today, Carola. Thank you so, so much for agreeing to give this talk.
Thank you so much, Elena. For me, it's also such a beautiful moment to be doing this with you. And I think it's a really nice reminder that in science, we not only make exciting discoveries, but we make wonderful friends. And I think I've had you very close for many years, and this is very special for me. So thank you, Elena, and you've been very generous as well.
Thank you so much. I value so much our friendship, Carola. And, you know, but still, we will ask a question that we ask all these global immune speakers, and because we would like the audience to get to know better the speakers from this seminar series. And the question we have for you today is, what is the advice you would give to your 25, 35-year-old self?
Right. Yeah, look, I thought when you said your 25-year-old self, I tried to think what I was doing at 25. And I was actually doing my medical residency in the UK. I had been 2 years doing junior and senior house officer jobs. And I remember I wasn't enjoying it at all. And I was really stressed about what to do. I mean, I'm just not enjoying something that I've been, preparing for such a long time. So of course I took a change in career, but I think looking back, I would tell anyone that is in my situation, if you're doing something you're not convinced, you don't enjoy, just follow your intuition, be courageous, life changes, try different things, and try until you find something that you really enjoy. And for me, it was science, what I discovered that I really enjoyed. There was insecurity, people were telling me that, I would get a worse salary, that I wouldn't have permanent jobs. But, I've never regretted that. And if any of you is out there with doubts, just keep trying, look for what makes you happy.
Thank you so much, Carola. This is a beautiful advice. Finding the courage to pursue what you're passionate about. Thank you so much. Okay, so we can proceed with your talk, and if you could share your screen.
Yes.
It works. Excellent. Perfect. Yes, we can see.
Great.
Okay.
Okay. So can you just remind me exactly how much time we have, Elena, from now?
From now, I would say, about 50 minutes. Is that okay? A few minutes more is okay to one. Sure, people will enjoy listening to you. A few minutes more is okay.
Okay. So I'm going to talk about, one of the areas that is closer to my heart, which are follicular T cells and the regulation of antibody responses and class switching. So first, just as a little bit of an introduction for those that are listening that might not be very familiar with B cell activation and B and T cell interactions. As you know, when a B cell gets activated, it needs to receive two signals. Signal one is classically a signal that is delivered when the antigen binds the B cell receptor. And signal two is typically delivered in the context of protein immunization or T-dependent responses. Is delivered by a T cell, and it tends to come in the form of CD40 ligand interactions and cytokines. So we now know that it is T follicular helper or TFH cells that are the specialized B cell helpers. And I think this notion was cemented when three groups, including my own in an effort led by DU, discovered the transcriptional regulator of TFH cells, or one of the key transcriptional regulators, which is BCL6. So BCL6 is important for many different aspects of TFH cell function, but importantly for orchestrating migration towards and into the follicles. We now know that there is quite a few molecules that are important in TB interactions. The chemokine receptor CXCR5 is crucial for aiding migration towards CXCL13 rich regions. We know that help is delivered through interactions of CD40 ligand with its receptor CD40, and there is a very interesting feed-forward loop mediated by ICOS and ICOS ligand that Haiji has described that is important too for this B cell and T cell communication. And we know that one of the key cytokines that TFH cells use to deliver B cell help is IL-21. So in nice work done by Michelle Lintemann in my lab and also David Tarlington's lab some years ago, we described how IL-21 acts intrinsically in B cells to boost germinal center B cell reactions. So just as a very brief overview of a typical TB cell interaction, first B cells get activated at the TB borders when they bind antigen, and they established a cognate interaction with the T follicular helper cells that have been previously primed during contact with an antigen presenting cell or a dendritic cell. So after the B cell has been primed in this first interaction with a TFH cell, it can take two routes of differentiation, and this is a simplified version of what really happens. It can differentiate in extrafollicular areas, giving rise to relatively low affinity, short-lived plasma cells, or it can differentiate forming germinal centers in follicles. So after a first round of division, the B cells move to the area of the follicle closer to the T zone, and here they divide rapidly and acquire somatic mutations in their immunoglobulin B region genes. Now, these mutations can change the affinity of the immunoglobulin. They can become higher affinity, even they could become self-reactive, or they can lose affinity altogether. So a selection process takes place by which B cells pick up antigen held by immune complexes on the surface of follicular dendritic cells, and these B cells process antigen and present it to T follicular helper cells. There is a competitive mechanism by which those B cells that present more antigen, which of course is a sign of being higher affinity, get predominantly T cell help. And those that do not get help die by apoptosis in the follicle. Now, the outcome of this interaction is that there is formation now of long-lived memory B cells or long-lived plasma cells that tend to migrate, amongst other places, to the bone marrow, where they can keep producing immunoglobulin for a long time. And this is now high affinity antibody. Now, we now know that this high affinity antibody produced by these long-lived plasma cells is quite important for protection. And a nice example, for example, is the antibody that gets produced against a vaccine like tetanus. In the context of tetanus, there is a neurotoxin that binds the presynaptic membrane at the neuromuscular junction. And if there has been antibody produced that is high affinity, the antibody can compete with the neurotoxin for binding the receptor. This antibody can be passed through the placenta. So if mothers are immunized, the children should not develop this very severe disease that still kills a very large proportion of children in the developing world. And you can see how the introduction of the vaccine has dramatically reduced mortality. So long-lived antibodies, those that are high affinity and can be transmitted through the placenta, which requires switching to IgG, are important. So we've now mentioned two concepts that are important in antibody diversification: acquisition of mutations that increase affinity for the immunizing antigen. This is the process of somatic hypermutation and affects mainly the variable region genes of the immunoglobulin, or class switch recombination that this time predominantly affects or changes the Fc fraction of the antibody that allows for different effector functions of antibodies. So we're going to focus on this part for the next little while. And why is switching important? Well, each of the isotypes can confer specific and important functions. We know that IgM is important in early host defense. It has high valency, can form pentamers, which is good at fixing complement. IgA is secreted into body fluids, and this is important to protect at mucosal surfaces. IgD is the form of the membrane receptor. IgE can bind to specific receptors on mast cells and trigger the production of histamine and other mediators of allergy. But of course, these cells are also important in defense against helminths. And IgG is quite potent in opsonization by binding Fc gamma receptors on different cell types, including macrophages. IgG is also important for antibody-dependent cell-mediated cytotoxicity. It can fix complement to different degrees depending on the different IgG subclasses. And as I've just mentioned, it's important for passive immunity in neonates because it can cross the placenta. So how and where does class switching occur? Well, this is the textbook view of germinal centers, where somatic mutations, I've just told you, occurs during the rapid B cell division that occurs in the dark zone of germinal centers. And it has been thought for quite a while that class switch recombination is favored or occurs predominantly in the light zone of germinal centers. Now, it is important to remember that there are certain pathologies that have been associated with defects in the process of class switch recombination. So not only there is a possibility of lymphomagenesis through the introduction of chromosomal translocations during the process of class switch recombination, but also there are other processes like insertion of introns that can occur during class switch recombination that in some cases can confer, for example, self-reactivity, and there's been some nice illustration of these effects by Antonio Lanzavecchia. Of course, some of these introns can also be quite protective against some infections, such as malaria. So just for a very brief overview that is important to the methodology that we're going to use, I'll just quickly tell you how the process of class switch recombination works. So class switch recombination relies on the activation of three key enzymes, AID, UNG, and APE1. And these enzymes together bring about DNA breaks that lead to recombination of a particular PDJ region with any one of the different possible constant heavy chains. Now, the selection of isotypes, which of these heavy chains is going to be chosen, is dependent on T cell help, the nature of T cell help, and the cytokines that are produced at the time of help. So it is these cytokines that induce transcription across this, what we call switch regions. Switch regions are intronic elements that are targeted by these particular sets of enzymes. So these switch transcripts, they are also called germline transcripts. They do not encode for protein. They are non-coding RNAs, and they are transcribed upstream of promoters that are found immediately upstream of these switch regions. Now, we know that expression of these switch transcripts precedes DNA recombination, And therefore, they have been considered very reliable markers to study the onset of class switch recombination. And after this process has taken place, now there is now the synthesis of a transcript that now has excised the intervening elements, and where the desired constant heavy chain region is now juxtaposed to the VDJ segment. So what is the evidence that class switch recombination occurs in germinal centers? Well, we know from old studies that there is a switch from what was thought to be, from IgM to what was thought to be a marker of class switch recombination, having switched to IgG, from the primary to the secondary response. So it was thought that the secondary response occurs mainly as a consequence of germinal center reaction. There is less class switch recombination in mice lacking germinal centers, but of course, many of these are genetically manipulated mouse models that not only lack germinal centers, but where TV demarcation is altered and there might not be productive TV cell interactions. And then there was the discovery by Honjo in 2000 of AID, this enzyme that is essential for class switch recombination and somatic hypermutation. And it was clear that in these mice, not only somatic hypermutation was drastically reduced, but there was less class switch recombination. Now, because AID can be prominently seen forming clusters in germinal centers, it was assumed that this was the location where class switch recombination occurred. Also, there was a paper that pinpointed the stage of class switch recombination as the centrocyte stage in germinal center cells in humans. And for this, there was a combination of markers used that was absence of CD77 in the presence of markers of B cell activation. But we now know that these are not the best markers to identify centrocytes, because indeed lack of CD77 identifies what is now known to be a heterogeneous subset of cells that not only comprise the centrocytes, but also activated B cells and plasmablasts. So there has been also some evidence that class switch recombination can occur before germinal center formation, and I'm citing 3 different studies. One came from the group of Ian McLennan with Kai-Michael Toellner, who showed that they could detect switch transcripts in total tonsil sections at a timing that was probably preceding the onset of a germinal center reaction. Now, Mark Jenkins also showed very nicely that he could identify switched IgG2A cells as early as 2 days after prime in a T-dependent response. And Rob Brink showed in 2009 parallel and comparable appearance of IgG in germinal centers and extrafollicular plasmablasts. So our question was, we've seen there is evidence that class switch recombination can occur prior to the germinal center, but does still class switch recombination predominantly occur in germinal centers? So to do this, we first used the switch health system that Rob Brink had described. This is quite useful to track antigen-specific B cells that bind HEL and can still undergo class switch recombination and somatic hypermutation, because these B cells carry an opt-in rearranged VDJ region that binds HEL with high affinity. Now, in order to have a lower affinity for antigen, a mutated form of HEL has been used and has been conjugated to sheep red blood cells, so there is unlimited T cell help. So what we did is transfer these switch HEL B cells into recipient black 6 mice and look at very early time points after immunization with either 12 hours or 24 hours apart for the first few days into the formation of germinal centers. And you can see that we can very easily distinguish pretty earlier on 2 populations that bear different markers, and we know we can separate very clearly germinal centers for the extrafollicular plasmablasts, and we've done this with every possible combination of markers, and we can see that by day 3.5, we start seeing the 2 populations, but prior to day 3.5, we still have a homogeneous population of activated B cells that haven't yet committed to one or the other fate. And this similar early appearance of germinal centers by day 3.5 can be confirmed by immunohistology. So in the first few days, we can up to day 3, we can only see hell-binding B cells here in red in the follicles. And by day 3.5, we start seeing the appearance of early germinal centers, where they are very clear now by day 4.5. And as you can see, at day 4.5, we clearly have both an extrafollicular plasmablast response and a germinal center reaction. So here, what I want to show you is what happens when are the switch transcripts produced. And I told you this is the best marker for the onset of class switch recombination. If we look at gamma-1, the first switch, the switch transcripts peak by day 3. And I've just told you that by day 3, there's still no germinal center formation. Here in the gray shaded areas, we see the germinal center process. So by the time germinal centers form, day 3.5, there's already been a decline in gamma-1 switch transcript. Now we see a very similar pattern for gamma-2b. The peak occurs between day 0.5 and day 3. And by day 3.5, we already see a decline of germline transcripts that continues throughout the germinal center reaction. So this suggests that class switch recombination is triggered prior to the germinal center reaction, and there doesn't seem to be much class switch recombination activity during germinal center reactions. Now, if we look at when is AID, or encoded by AICDA, when is it first induced? Well, it is induced as early as day 2.5, at the time when we are seeing germline transcripts produced, and then there is further expression in germinal centers. So presumably this amount of AID could drive germline transcription. Sorry, not germline transcription, but could already be acting up to promote class switch recombination. And what about BCL6? Well, as I've just told you, and consistent with the lack of germinal centers on day 3, there is no BCL6 expression on day 3. And remember that BCL6 is the transcriptional factor of germinal center B cells. And by day 3.5, we now have BCL6 expression coincided with the expression of germinal centers. So from this experiment, it looks as if class switch recombination would be triggered prior to the onset of germinal centers, with very little evidence that it continues. So we could argue that this class switch recombination in the germinal center might be occurring in a subset of light zone cells that we are missing by doing this bulk sequencing of germinal center B cells. So what we did here, and this work was done by Jonathan Rocco in collaboration with Jose Polo, what he did is sort cells, hell-binding B cells at day 3, when I've told you this is prior to germinal center formation, we are calling these B blasts, or day 6.5, separating dark zone and light zone subsets with the markers that Gabriel Victoria described, or at day 6.5, same time point, but light zone cells. And we sequence up to 100 different light zone cells, we performed real-time PCR for a number of markers, including all switch transcripts. And what emerged from this exercise is that most of the gamma-1 and gamma-2b switch transcripts are produced early on day 3. If we sort, as I say, amongst 42 dark zone cells and 100 light zone cells, we found very rare events of switch transcripts. We do see some, so it's not 0, but it is a minority of switch transcription events. And here you can see that we have AID expression, both in these day 3 B blasts and in germinal center B cells. And you can see that BCL6 is not, or is barely expressed at day 3, but is prominently expressed in these other cells. So this again supports the notion that most of the switch transcript or class switch recombination events are triggered prior to the germinal center. So we also asked, could we physically visualize where class switch recombination is being triggered? So for this, we used these mice in which when the gamma-1 germline transcript is induced, the B cells switch from being red to being green. So what we did is transfer these B cells into mice, immunize the mice, and ask, when do we first see green cells, so cells that have expressed the germline transcript, and where are they? So as early as day 2, and this work was done in collaboration with Kai-Michael Troellner and Yang Zhang, we could see that the first B cells are seen at the TB border at day 2 prior to the onset of germinal center formation, suggesting that class switch recombination is occurring very early and in the absence of germinal center formation. And this, you can see that by day 3, still there are no germinal centers, but we see many more of these green cells entering follicles and already starting some of them to localize as extrafollicular plasmablasts. Now by day 4, germinal centers have formed, and of course these cells have now divided and are prominent in germinal centers and in the T zones. And again, we now see switch cells as abundant in both locations. All of this again suggesting that the first events occurred prior to the onset of germinal center formation. So can we also look at the protein? When do we see the immunoglobulin isotype on the surface of B cells, not just the germline transcript? So in the health switch system, we saw the first IgG1 B cells appear as early as day 2.5, again suggesting this is occurring prior to germinal center formation. And if we look at the kinetics of the appearance of extrafollicular plasmablasts and germinal centers, we now see that the proportion of switch cells in these two compartments is completely comparable from day 4.5, with no suggestion that there is more switching occurring in germinal centers. In fact, if anything, in later time points, we see a higher frequency of switch cells in the extrafollicular plasmablast response. Now, if we now look at the kinetics of the germinal centers, we see that the germinal centers peak more or less from day 8. And if we now enumerate the IgG cells in these germinal centers, we can see that these have peaked also at day 8, and there is no further increase of switch cells, suggesting again that this is not an ongoing process in germinal centers. So all of this so far has been done using a transgenic B cells, and we did use other models that I'm not presenting here. So first we wanted to see, can we show the same thing in a polyclonal repertoire, but also more importantly, can we identify the timing of class switch recombination relative to somatic hypermutation? So which occurs first? And here we started a very exciting collaboration with Luca Messing and Gabriel Victora. So they used their photoactivable system in which you can photoactivate a particular germinal center in a lymph node. These cells are then marked in green, so these cells can now be sorted, and we can sequence both the VDJ region to look for somatic hypermutation and the constant heavy chain region to look for the switched isotype. So this is a graphic representation of the number of germinal centers that were sorted and sequenced, and this experiment was performed at 2 time points, day 15 and day 20. So we focused the analysis on those germinal centers that were enriched for clones in which they had more than 4 IgM cells. Each of these boxes represents a clone. In green there is IgM, and in gray there is IgG, with the assumption that the more IgM cells, the more opportunities to see switching, and every box or every clone that contained both IgM and IgG cells were sequenced to see when this class switch recombination had occurred. Just to show you how the analysis is displayed, in white we have IgM and switch cells. In colors we have switched cells. IgG1 is shown in black. Now the red triangle shows the point at which class switch recombination event has occurred. If the triangle is open, means it is a clone without class switch recombination. And on the x-axis, we are displaying the number of somatic mutations. So 0 somatic mutations to 10 mutations, for example, for this particular cell. So what do the results look like? Well, you can see that if we look at where the red triangles are, nearly all of the class switch recombination events have occurred either at 0 somatic mutations, with the 1 or 2 exceptions. Here it occurs at 1 and at 4, but every other instance we see either unswitched clones that remain unswitched throughout the entire germinal center process or after acquiring up to 10 mutations, or there is a switching event but no further class switch recombinations occurring. And this is now enumerated here. So the conclusion of this experiment is that class switch recombination occurs in unmutated cells predominantly, with some exceptions. So now the other interesting observation is that from this diagram, you can see that there are germinal centers like this number 3 or this number 5 that are predominantly IgM, that are dominated by IgM. Now, we've done some modeling work with Michael Meyer-Herman, but even without the modeling, you can imagine that there must be a negligible probability of IgM-dominated germinal centers in the if there is constant switching in germinal centers. So again, these findings suggest that class switch recombination is not a prominent process in the germinal center. So the other interesting thing, trying to see how this could be regulated, I've already shown you that a level of regulation is clearly at the germline transcription. There is less germline transcription occurring once a B cell becomes a germinal center B cells. But the other interesting finding is that this enzyme APE1, which has been shown to be important for class switch recombination, and these are some experiments in cell lines, it's downregulated in germinal center B cells. And we showed this both at the transcript level and at the protein level. Now, in mice, there was a previous report of lower APE1 expression in germinal centers, and we confirmed that in humans in both dark zone and light zone. And you can see it very clearly here by immunofluorescence that there is generally very little APE1 expression in germinal centers, whereas APE2, which is the enzyme that's most important for somatic hypermutation, is very prominently expressed in germinal centers. And of note, we reanalyzed some data that Jose Polo had produced doing chip-on-chip on human primary germinal center B cells, and found that APE1 is a target of BCL6. And BCL6 is a transcriptional repressor, so this offers a mechanism by which BCL6 might turn down transcription of APE1. This is APE2 as a control, and you can see that this is not the case for APE2. So this was published last year in Immunity, and this is the work, as I say, of Jonathan Rocco with many collaborators that I have mentioned. And I think the take-home message is that class switch recombination occurs first during the first TB-commit interaction at the TB border prior to entering germinal centers, and in germinal centers, somatic hypermutation mainly occurs. Now that doesn't mean that a cell that has exited the germinal center, like a memory cell, cannot undergo another process of class switch recombination. But again, we anticipate this would occur outside of a germinal center upon contact with another T cell, probably a memory T cell. Now we could think, why would there be an advantage at not having class switch recombination in the germinal center? And I think part of the answer could come from the need to maintain diversity in the memory compartment. It would be important that memory cells can be reactivating by variant forms of antigen upon subsequent encounter or mutated viruses. And this increased diversity of preserving an IgM unmutated memory pool, or not unmutated, sorry, unswitched, could allow for switching to different isotypes that can be more protective in the presence of a variant form of the original antigen. So with this, I'm going to now go to the second part of the talk. We're still going to talk about antibodies, but this time the regulation of antibodies that could be pathogenic, in particular the regulation of IgE. And we know that IgE is an isotype that mediates allergic reaction to a number of allergens and food proteins through its ability to trigger mast cell degranulation through binding of the Fc to Fc epsilon receptors in mast cells. Now, I'm first now going to tell you or remind you about a cell subset that 3 groups, including our group, in an effort led by Michel Lintemann, described some years ago, which are these T follicular regulatory cells. So we now know that thymic-derived Tregs, and there is some evidence that possibly also induced Tregs, can co-opt the TFH differentiation pathway, express BCL6, upregulate CXCR5, maintain expression of FOXP3, and therefore migrate to the TB borders and enter also possibly germinal centers to regulate B cell responses. And it's been a little bit less clear what these TFR cells or T follicular regulatory cells look like in humans. So one of the most accessible tissues in humans are tonsils. This particular tissue is probably quite relevant in terms of tolerization to both allergens and innocuous inhaled microbes, because as you see, it's located in a site that is continuously exposed to inhaled and digested food antigens. So if we look at what these tonsils look like, and they don't look that different to lymph nodes, but there are some differences apart from chronic antigen exposure, we can see a very clear compartment of what we defined as germinal center follicular T cells. They are very high in PD-1 and CXCR5. Then this intermediate compartment that contains other effector T cells, and these less activated cells that can be enriched in naive cells. If we now do the typical gating for human regulatory T cells, which consists on expression of CD25 in the absence of CD127, we can see the typical Treg population amongst this effector T cell subset, very few Tregs amongst the more naive T cells, but we clearly see what could look or do look like Tregs amongst these follicular T cells. So in principle, we would have called these T follicular regulatory cells. Now, what we were surprised by, and this is work done by Pablo in my lab, what we were surprised by is that these CD25 positive follicular T cells, and we are calling them these throughout the talk, CD25 positive TF, unlike Tregs, barely express FOXP3. And this is something quite unique to the tonsil. It's highly enriched in these T cells that express very little FOXP3, both as a proportion of total amount of these cells, but also in MF5. So do they look like Tregs? Well, first we looked at the methylation of the CNS2 promoter. We know that thymic-derived Tregs have a demethylated CNS2 locus, and this is important for the stable expression of FOXP3. And we can see that whereas Tregs are generally demethylated in 3 different donors, our CD25 positive TF cells look methylated. So if they are Tregs, they don't look like a typical thymic-derived Tregs. Now, what about transcription? Well, first of all, you can see that the CD25 positive follicular T cells do not express LAG3, so they are not the TR1 or induced T cells, Tregs, but in everything else, they look very similar to Tregs. They have very high expression of CTLA4, high expression of SKITAR, low expression of CD40 ligand, very high expression of IL-21. They do express IL-21 and this, sorry, IL-10, they do express IL-21 and this is different. Now, what about at the protein level? So we've confirmed the transcription pattern at the protein level. And here we've included Tregs in blue. So our CD25 positive TF cells in red actually express less CD40 ligand compared to TFH cells in green, but express the highest amounts of CTLA4, even higher amounts than conventional Tregs. They express the highest amounts of ITAR, co-express BLIMP1 and BCL6, and also don't express very high amounts of the IL-10 receptor, but do express IL-21, and again, this could be important. So if they look like Tregs, but they don't express FOXP3, can they act like Tregs? Can they suppress proliferation of T cells in vitro? So what we found is that they act like Tregs. Indeed, they are as suppressive, and this is our CD25 positive T cells in the filled circles. They are as suppressive as conventional Tregs in an in vitro suppression assay, whereas, for example, conventional TFH cells cannot suppress. So these are not just activated TFH cells, these look like suppressive T cells. And the other interesting thing is that, at least in tonsil, these CD25 positive T follicular cells are the highest expressors of IL-10. You can see Tregs can also express some IL-10, but in the follicles, these are the highest expressors. So we asked, do they regulate antibody production? And we found that our CD25 positive TF cells do not do much in terms of regulation of IgG, but they themselves cannot induce IgE, and in cultures in which there are TFH cells, they repress production of IgE that would normally be induced by these TFH cells. So it looks as if like the CD25 T follicular cells are particularly good at suppressing IgE in human tonsil cultures. So how do they suppress IgE? Well, what we found is that they act at the very first stage at the induction of epsilon germline transcripts. So here you have cultures that in which we have not added these CD25 positive follicular T cells versus addition, and you can see that generally there is less epsilon transcripts when we've added T follicular cells, and we don't get the same picture for gamma transcripts. So this particularly affects switching to IgE. And the question is, how is this mediated? Could IL-10 that we've seen is produced predominantly by this T follicular subset be important? Well, if we now block IL-10 by the introduction of an anti-IL-10 receptor, we rescue repression of the epsilon germline transcript. And again, this doesn't do anything for IgG. So is this relevant to human physiology? So what Pablo did is an exercise of analyzing 50 children and taking the serum from the children and comparing the amount of these follicular CD25 positive T cells in the tonsil of each child. So these are correlations between serum IgE in one child and the amount of CD25 T cells, either as T follicular cells in the tonsil, either as a proportion of Tregs or a proportion of total CD4s. And you can see that there is a very nice inverse correlation, so that those children that have higher IgE in the serum tend to be those that have a lower fraction of the CD25 positive TF cells. And if we now ask, does this have to do with the IL-10 producing subset? And it looks like it does, because there is now also an inverse correlation between those children that have high serum titers of IgE, which tend to be those that have a lower fraction of IL-10 producing follicular T cells. And we did not see that correlation between total FOXP3 positive cells in the follicles, which I told you are very rare, but we could still enumerate them, or total IL-10 producing T cells or Tregs, suggesting that this is a specific function of these CD25 positive TF cells. And another way of looking at it, if we now divide children into children that are high IgE, and this is a threshold that is used commonly in the clinic, children that have more than 100 nanograms per ml, shown in the open circles, or children that have low IgE, and you can see that those children that have high IgE tend to be those that have a low fraction of CD25 positive TF cells, either again as a proportion of Tregs or of T cells. So we think this is important, and this is a summary of this part of the talk. We can see that in the tonsil there are these CD25 positive TF cells that secrete IL-10. Haven't told you about this part of the data, but Pablo Cañete also showed that these cells not only act on B cells, they can also act to dampen TFH cell health by reducing BCL6, CD40 ligand expression, IL-21, and they also, as I've just shown you, repress class switch recombination to IgE so that there is less serum IgE produced by plasma cells. So we are very happy, and I think it's really nice for Pablo that his paper was selected as one of the top 10 papers in JXMed last year. And I'm left with now thanking my entire group. I've mentioned Jonathan, who performed most of the work in the class switch recombination project, class switch recombination not occurring in germinal centers. Here is Pablo, who has performed this work on the human CD25 positive follicular T cells, and these are all of the other members of my lab. Of course, there is quite a lot of other people that have contributed, and I think I have mentioned all of them as we were going ahead. And before we finish, I would also like to mention my China Renji Hospital lab in Shanghai. So watch this space, because we are now investigating the genetic causes of sporadic pediatric autoimmunity, and there is some really nice findings that hopefully, if I'm ever invited again in the future to another one of these talks, you will hear some really nice findings. So with this, I would like to end my talk. Thank you very much for listening.
Carola, thank you so much. That was beautiful. Both stories, very enlightening. Yes, thank you. It's always a pleasure to hear you. So thanks a lot for your effort to prepare the talk and for sharing your great research with us.
Thank you very much, Elena, and thanks to everybody for being here. Thanks to my lab for doing really beautiful work.
Fantastic. So you agree then questions will be via Twitter, and so whenever you are available, we will direct the attendees to ask questions via Twitter, and yes, you can answer at whenever you have time to do so.
Thank you very much, Elena, and I'm not very good with Twitter, but I'll try my best.
Thank you, Carola. I appreciate it.
Okay,
bye everyone.
So thank you, everybody. And let's see. Elena just mentioned that Carola will be answering her questions via Twitter. So you should already be able to find this tweet that says ask questions for Dr. Carola Vinuesa here. And just remember, when you ask your question, please add the hashtag #GlobalImmuno. And today, Carola is going to be using her own Twitter account, which is @CaroVinuesa. So thanks again, everybody. And we look forward to seeing you again next Wednesday when we will have John Kapler joining us. See you soon. Bye-bye.
Bye.